So welcome back again. If you stepped away and took a break, or if you continued on with us, whichever way you went, welcome back again. Let's take a look at these concept questions, concept check questions, and then talk about the last little bit here. So what is the source of the potential energy that drives the ATP synthase that makes ATP? Yes, we could talk about the electrons, but remember the energy of the electrons is converted to a hydrogen ion gradient. So it's the hydrogen ion gradient. Let's go over here and do it over here. So remember again, it's the energy that's extracted out of the electrons in the electron transport chain that generates the hydrogen ion gradient. And it's that hydrogen ion gradient that actually provides the energy for driving the ATP synthase. Which of the three stages of cell respiration yields the most ATP? Well, if you say the electron transport process, you would be correct, because that's how the cell describes it. But if you want the more complete term, it would be oxidative phosphorylation. That would be the more correct term. But if you think in terms of electron transport process, you'll be fine as far as exam questions will be concerned. So those are the answers to these two top concept check questions. So one more section, let's get to it so that we can finish this up and tidy up this presentation. So fermentation, why does fermentation exist? Well, some of you may be thinking so that we can have adult beverages. And while that is the product or a product of fermentation, it's not the reason why fermentation exists. No, the yeasts were not thinking about human pleasure and human enjoyment when they decided to, or they evolved the process of fermentation. And in fact, fermentation evolved long before there were yeasts. Fermentation evolved long before there were anaerobic organisms, long before there was even oxygen in the atmosphere, as it turns out. Fermentation is a way of getting around a fundamental problem with glycolysis. Because if you are anaerobic and you're carrying out cell respiration, you need oxygen. But if you are anaerobic, which means you're functioning in the absence of oxygen, you have a fundamental problem. And it ties into glycolysis, which is your only source of ATP. It turns out that to make ATP in glycolysis, you first have to reduce NAD plus to NADH. So to make must first reduce NAD plus to NADH. The problem is that there is a finite supply of NAD plus to be reduced to NADH, which means you need to recycle the NADH back to NAD plus. In other words, you need to oxidize it back to NAD plus. That is normally taken care of by the function of the mitochondria. But the function of the mitochondria requires oxygen. So what if you don't have oxygen? Or what if you don't have mitochondria? How can you regenerate the NAD plus that you need to allow glycolysis to continue to function? That's where fermentation comes in. Fermentation So it's an alternative way to oxidize NADH to NAD plus to allow glycolysis to continue to function in the absence of oxygen. Because in the absence of oxygen, you don't have an electron transport chain, you don't have a functional mitochondria, you're not oxidizing NADH back to NAD+. Got to have an alternative way of doing it. That's the role of fermentation. So this animation walks you very quickly through the two basic types of fermentation, what is referred to as alcoholic fermentation, that's what yeasts do. And then the alternative common way of doing fermentation is what is referred to as lactate or lactic acid fermentation. That's what a large number of bacteria do. So when you think in terms of sour cream, yogurt, fermented milk products, basically, or anything that's pickled. So pickles, sauerkraut, kimchi, pepperoni, salami, those are all the product of lactic acid fermentation processes carried out by bacteria. So basically, it's a way of souring material or pickling material in a very, very controlled fashion. It also happens to be the temporary solution for muscle cells that go anaerobic. They also perform lactic acid fermentation, but it is very much temporary. So this animation is again available in the Chapter 6 resource page. And I would strongly recommend that you go and take a look at this animation because it gives you a very nice overview of the two different types of fermentation. It's not as pretty as the Bioflix animation, but it still gives you a really nice overview of the two different types of fermentation. We will talk about them here, but looking at the animation is often beneficial for if you like that sort of thing, if that's helpful. So let's take a look at the two different types of fermentation. And the whole point of saying that fermentation in muscle cells is temporary is just the simple recognition that most muscles, particularly the big muscles of the legs and the arms and your heart, can really only function anaerobically for about 15 seconds before things start to go wrong. They will begin to start generating ATP by fermentation, but remember, they're expecting bucket loads of ATP via aerobic respiration. When they have to resort to anaerobic respiration, they're not going to get those bucket loads of ATP. And that's going to reduce their efficiency, and eventually the muscle starts to break down, or you get a muscle cramp, depending on the type of muscle that's being affected. So heart muscle needs to keep working. reduce the amount of oxygen getting to heart muscle, you get death of heart muscle. We call it a heart attack. In skeletal muscles, so the big muscles of your arms and legs, typically those will cramp up rather than have muscle cells die. But that can even happen if you insist on forcing the issue and the cells can't switch over to an alternative form of fuel. So if they use up all of their fuel, their readily available fuel, and they're running low on oxygen, they may very well start to die themselves if they don't cramp up first. So it becomes a bit of an issue if you're not able to take care of this by transporting oxygen efficiently. That's why cardio exercise, aerobic exercise, is really important to build up your aerobic capacity, as we talked about at the beginning. So we've talked about all of this, the fact that we need to recycle the NAD+ in order to allow glycolysis to continue working. So here is the pathway that your cells follow. So it's going to look remarkably like glycolysis in aerobic respiration, except it's the fate of the pyruvate that changes. So here we have glycolysis occurring, and you can see that we're producing NAD, we're producing ATP, but in order to be able to do that, we need to be able to reduce NAD+ to NADH. That's the first step that must take place before you can make ATP. You've got to do the reduction of NAD+ before you can make the ATP. So we really do need NAD+ available. So we get the pyruvate, but instead of the pyruvate passing on into the mitochondria, it stays in the cytoplasm and it is itself reduced to lactic acid. But of course, if you do a reduction, you need an oxidation. You need a source of electrons. That means you need an oxidation. You can't have one without the other, as we keep talking about. So we use the electrons of NADH to reduce pyruvic acid to lactate, which accomplishes what fermentation is really all about, the oxidation of NADH back to NAD+, which gives us the source of NAD+ to allow glycolysis to continue. So this is found in muscles. And anaerobic bacteria. So there are some bacteria that carry out slightly different forms of fermentation. No bacteria does alcoholic fermentation. But there are some specialized forms of fermentation done by some bacteria, but you don't want to mess with those bacteria. Those are really, really dangerous bacteria. They cause food poisoning, and some of the more extreme types of these bacteria can actually kill you as a consequence of their types of fermentation and the byproducts that they make as part of their fermentation process. Ironically, one of those types of bacteria is something called Clostridium botulinum. And the byproduct of the botulinum bacterium in a diluted form is what is injected in Botox injections. And a Botox injection can actually kill you if it's given incorrectly, because you're actually using a bacterial toxin that's produced by one of these specialist fermenting bacteria. Again, these are the sort of bacteria you don't want to mess with. In the case of the botulinum bacterium, if you ever come across a a tin can that's swollen, that the ends are swollen, don't even bother opening it. That's simply an indication that something on the inside desperately wants to get out. And that usually means that the botulinum bacterium is growing in there, and it will kill you if you try and eat that food because of the food poisoning that it causes. They're really, really nasty bacteria. Leave them alone. Don't mess with them. So this is lactic acid fermentation, or what is referred to as lactate fermentation. This is what your muscles do. So back in the 1920s, research indicated that the buildup of lactic acid was what was actually causing muscle cramping. Now, you've got to be really careful about accepting this data, because what the research was done on frog muscles, and frog muscles don't work at the same temperature that our muscles work at. But people didn't realize that this was an issue when this research was done. So the experimental setup was a situation like this, where the frog muscles were extracted out of the frog's legs, connected up to this device, and then connected up to a battery. And when an electrical stimulus is applied to a muscle, it will contract. And so the two setups were a solution that contained lactic acid so that the lactic acid could not diffuse out of the muscle, and a lactic acid that had the solution that had no lactic acid in it, so that these lactic acid in the muscles could diffuse out of it. And in this setup, the muscles stopped working very quickly, whereas in this setup, the muscles continued to work for an extended period of time. And the conclusion drawn from this was that the presence of lactic acid in the muscle was what was stopping the muscle from working, was the cause of muscle cramp. There are a number of issues with this. One is that it was working with frog muscle that operates at a lower temperature than human muscle. So human muscle may not be as sensitive to the buildup of lactic acid. Number two is that there are individuals who have muscle cramps but no buildup of lactic acid. And in fact, their muscles cramp much quicker in the absence of the lactic acid than in individuals who have the buildup of lactic acid, which suggests that in fact, the lactic acid may be protecting against the muscle cramp. And then there are other research studies that suggest that there may be other things other than the lactic acid that could be triggering the muscle cramps, because the research suggests that the buildup of these other things, particularly some different types of ions in muscles, may be responsible for muscle cramp. So if you go and do research on muscle cramps and you ask the basic question, what's caused a muscle cramp? You're going to find people who are saying, well, it's the lactic acid. And then you're going to find people who are saying, no, it's not the lactic acid at all, it's something else. And that's because the research data is confusing. The original studies suggested that it was the lactic acid, but because of the issues with those original studies, we can't necessarily apply that data to human muscles. Plus, there is evidence now being developed from looking at human muscles that suggest that the lactic acid is not the culprit, that it's something else. More research is required in order to answer the question. And there are ongoing studies, but the preponderance of information these days suggests that the lactic acid is a symptom, not a cause of the cramping and the anaerobic behavior of the muscles. So when we start thinking in terms of bacteria and other microorganisms, as I said, lactic acid fermentation is a very common way for bacteria to do it. We take advantage of that in making fermented dairy products, so cheese, sour cream, yogurt, kefir. There are a couple of other fermented dairy drinks that I've forgotten the name of, but they're all the product of of bacteria. The reason why they have a slightly acidic bite is because of the presence of the lactic acid. Soy sauce is a fermented soybean product, and again, it's a lactic acid fermentation. Pickles, of course, are acidic, it's lactic acid. Cabbage, that's sauerkraut, and kimchi, olives. Sausage meat is kielbasa, pepperoni, salami. They are all preserved by external fermentation on the surface of the meat, and the buildup of salt and lactic acid that prevents the growth of bacteria in the meat product. It's a good way of preserving the sausage meat. In the case of yeasts, yeasts are the only organisms that can flip backwards and forwards between aerobic and anaerobic respiration and produce alcohol. So rather than doing the lactic acid fermentation, yeasts do alcoholic fermentation, but it allows them to be both aerobic and anaerobic depending on the conditions. So when there's when there's oxygen present, they are aerobic and they do not produce alcohol at all. But when they become anaerobic, they don't stop functioning, they simply switch over to alcoholic fermentation, which allows glycolysis to continue, but as a product of their form of fermentation, they make alcohol. And so if you want to make beer and wine at home, which you can in some locations, the one thing that you need to do to make sure that you actually make the alcohol is make sure that the containers that you're doing it in are airtight so that they consume the oxygen and go anaerobic in order to make the alcohol. Otherwise, what you're going to wind up with is a very, very sweet, non-alcoholic solution that will be very, very disappointing. But what you may not have realized is that anything that uses yeast as a leavening agent, so when you make bread and rolls and other things that use yeast as the leavening agent, the yeast there is also anaerobic. And the captured carbon dioxide in the gluten complex in the bread dough is what causes the dough to expand or to rise. And when you cut bread and you see those little bubble type pockets in the surface of the bread, that's where the carbon dioxide was that caused the bread dough to rise. It was captured inside the gluten complex and it expanded the bread dough. Do you make alcohol in the bread dough? Oh, yes, you do. But when you bake the bread, you bake the alcohol out of it. But nonetheless, you can influence the taste of the bread by using different types of yeast. So sourdough bread is actually using natural yeasts in the environment. When you make your sourdough starter, you take flour and mix it with water and you leave it exposed to air, and that captures the yeasts that are in the air around you, and those start to ferment the carbohydrates in the flour, and that gives you the sourdough starter. And apparently the native yeasts in San Francisco provide the best sourdough starter, and that's why San Francisco sourdough bread is considered to be the best sourdough bread because of the nature of the yeasts, which are natural and native to San Francisco. But you can make your own sourdough starter locally. It's just a way of capturing native yeast. And the same is true with making particularly wines. The reason why different wines in different locations have slightly different qualities is because they're relying on the natural yeasts that sit on the surface of the grapes to produce the alcohol through fermentation, and the different types of regional yeasts give different properties to the different types of wines. Even if it's the same type of wine using the same type of grapes, but grown in two different locations and fermented in two different locations, you will get different properties simply because of the different types of yeast. And it could be even from one valley versus the next valley, or one side of the valley versus the other side of the valley. It is very regional in terms of the type of yeasts that are available. So what does alcoholic fermentation look like? Again, it looks very similar to what goes on in aerobic respiration initially. So glycolysis takes place, you get the pyruvate. You even get the removal of the carbon dioxide to get acetyl groups. So you get what is referred to as acid aldehyde, which then gets converted to ethyl alcohol. So even the acid aldehyde is very similar to the acetyl group, but not identical, not chemically identical. It's very similar to it though. And it's the acid aldehyde that then gets reduced to ethyl alcohol. But of course, if you do a reduction, you've got to have an oxidation. And that means that the electrons from NADH that carry out the reduction of the acid aldehyde to alcohol also leave the NADH now in its oxidized form NAD+, which allows glycolysis to continue. What is quite remarkable here is that the yeasts are carrying out a process when they do this fermentation process that will result in a substance, the alcohol, that will kill them. Because remember, alcohol is a cellular toxin, and it's just as toxic to the yeast as it is to anything else. But they are so dependent on the ATP from glycolysis, they carry out this process that will ultimately lead to their death if it produces sufficient alcohol. I think nothing more than this simple observation reinforces just how important the ability to make ATP actually is to cellular survival. That yeast will carry out a process that generates the ATP that they need, but that will also ultimately wind up in their death if they pursue the production of ethyl alcohol. Kind of interesting when you think about it from that perspective. So just how important is oxygen? Well, it turns out that oxygen and its availability triggers a split point in aerobic and anaerobic respiration. And pyruvate and the fate of pyruvate represents that split point in the two pathways. If oxygen is present, then the pyruvate enters the mitochondria or enters the aerobic portion of cell respiration in aerobic bacteria, and you process the pyruvate through the citric acid cycle and the electron transport process, and you get the boatload of ATP that you expect. If, on the other hand, the bacterium is anaerobic, or you're a yeast or a muscle cell that lacks sufficient oxygen, then you become anaerobic. In the case of the yeast, no harm, no foul. You continue to grow even though you have alcohol present. In the case of the muscles, it's temporary. But what we need to remember is that glycolysis, as that universal non-oxygen requiring part of cell respiration, also gives us some clues as to the origin of cell respiration and the ability to generate ATP chemically. Because it lets us know that this must be a really, really ancient process that evolved very, very early in the history of life on the planet, because the very, very earliest fossils that we have of cells are of cells that are capable of photosynthesis. We call them cyanobacteria. They're about 3.5 billion years old. If you want the exact time, it's 3.48 billion years. And that tells us that at that point, life was starting to produce oxygen. But prior to that, there was probably very little, if any, oxygen present in the environment. And it wouldn't be until around 2.5 billion years ago that there would be sufficient oxygen to start generating an oxygen-containing atmosphere that we would recognize as being similar to our own. Yes, the oxygen levels were significantly lower than ours. We have about 20% oxygen. but there would nonetheless be sufficient oxygen to provide a basis for the evolution of an aerobic form of respiration. But we know that there must have been several intermediates along the way, including glycolysis, but also some forms of anaerobic respiration that we still find present in some bacteria today, where rather than using oxygen as the final acceptor of electrons from the electron transport chain, you use other molecules. And one that you may have encountered is a form of anaerobic respiration that uses sulfur dioxide as the final acceptor of electrons. And ultimately that produces a gas called H2S, hydrogen sulfide, which looks a lot like water. Remember, water is H2O, hydrogen sulfide is H2S. And the pathway to it is very similar, but you use the sulfur dioxide as the recipient of the electrons, and H2S is the product. And H2S, or hydrogen sulfide, is rotten egg gas. It's sometimes also referred to as swamp gas or sewer gas. So if you walk through a swamp, you may smell this sulfurous rotten egg-like smell. That's because these bacteria are present. Or if you have a drain dry out, because the water trapped in the S bend of a drain is to stop this sewer gas from coming back up the drain. So if you have a drain that dries out and you don't have sufficient water to trap the gas, you might smell a rotten egg gas smell. So these are alternative forms of respiration. And while they're both called anaerobic respiration, they are different forms of anaerobic respiration to separate them from aerobic respiration that involves oxygen. These are types of respiration that occur in the absence of oxygen, either glycolysis in combination with fermentation, or a form of anaerobic respiration that uses the same chemical pathways as aerobic respiration, but the electrons wind up in a different type of molecule. So oxygen itself has become an important part of the ability to generate large amounts of energy in the form of ATP. So this just gives you a timeline as to what we think is the progressive development of an oxygen-based atmosphere that would eventually allow for the appearance of aerobic organisms as we would recognize them, first in the form of aerobic bacteria, but then ultimately aerobic eukaryotic organisms, because remember again, the mitochondria, where the aerobic portion of cell respiration occurs in eukaryotes, is itself derived through endosymbiosis from an aerobic bacterium. So all roads lead back to aerobic bacteria, and then ultimately back to anaerobic bacteria as a source of glycolysis. So one more concept check before we close up this presentation. Compared to aerobic respiration, how many ATP molecules are produced by fermentation per glucose molecule? Well, remember again, aerobic respiration gives rise to approximately 32 ATP. Anaerobic, and here I'm going to put in parentheses, glycolysis, only produces 2 ATP. And so even if we add fermentation, it's still going to be 2 ATP. So you can see why aerobic organisms really want to have access to oxygen, because the alternative is not going to work out so well in terms of the production of ATP. But for those organisms that can only do anaerobic respiration, again, a small amount of ATP is a lot better than no ATP at all, because in the absence of ATP, you're dead. So even the small amount of ATP that comes from anaerobic respiration is better than none at all. So that's cell respiration and a somewhat in-depth analysis of the process and the inputs and outputs and the alternative ways of generating energy. So, as always, if you have any comments, concerns, or questions about anything that I've covered in these animations, by all means, come and see me during office hours. Otherwise, I will see you again when we meet for the next narrated presentation, when we'll be covering Chapter 7, photosynthesis, the partner biochemical pathway for cell respiration. Bye till then.